Hello students, today we will be discussing about introns and exons and reverse transcription in two separate topics. Let's first discuss about introns and exons. In introduction, in the 1960s, non-bacterial, that is eukaryotic ribosomal RNAs were found to be synthesized as a long precursor RNA which was subsequently processed by the removal of apparently functionless internal spacer sequences. Since bacterial or prokaryotic ribosomal RNAs were more compactly organized, it was reasonable to ask whether the first ribosomal RNAs to evolve had the spacer sequences, which subsequently decreased in prokaryotes, or whether the spacer sequences were later acquired in the eukaryotes. Similar processing was found to apply to eukaryotic precursor messenger mRNAs. In the mid-1970s, it was found that some of the internal sequences interrupted the protein encoding part of the corresponding mRNAs. The internal sequences which were removed were named introns, and what remained in the processed mRNA constituted the exons. Let us discuss more about functions of introns. In the first function, regulation of alternative splicing, introns are crucial because the protein variety is greatly enhanced by alternative splicing, in which introns take part important roles. Alternative splicing is a controlled molecular mechanism producing multiple variant protein from a single gene in eukaryotic cell. It has been noticed that introns carry cis-acting elements that regulate alternative splicing. In fact, short cis-acting motifs that are necessary for binding splicing factors have been recognized and named intronic splicing silencers and intronic splicing enhancers. In the next function, positive regulation of gene expression, the expression enhancing effect of introns was first recognized in the experiment using simian virus 40 construct with or without introns, showing that their protein product was significantly diminished without their introns. Subsequently, Buckman and Berg showed that in a certain condition, constructs with introns were expressed up to 400 times higher than construct without introns, suggesting that introns can strongly enhance gene expression. Classically, enhancers mediate either direction of expression, up and down regulation of genes, and involve both spatial and temporal control of gene expression in a specific cell dependent of genomic location. On the contrary, intron-mediated enhancers, mainly identified in plant, generally act in the expression enhancement of genes and are primarily located in the first ordinary intron position within a gene. Transcription initiation and termination processes are cellular processes that involve introns as well, which need some sequence elements in introns to be correctly completed. For instance, some studies showed that specific sequence element in introns such as enhancers and silencers regulate transcription initiation through modulating the function of the promoters of genes. Introns also function as regulation of nonsense mediated decay. Nonsense mediated decay, also known as NMD, was originally known as a surveillance mechanism in eukaryotes that selectively removes mRNAs containing erroneously generated premature termination codons. However, several recent studies have suggested that NMD may be another normal mechanism of post-transcriptional gene expression regulation. Consistently, A recent study has shown that the levels of the expression of genes important for plant development are regulated by NMD. Generally, NMD recognizes the transcript on which an exon-exon junction complex, also known as EJC, resides more than 50 to 55 base pairs downstream of an authentic termination codon as the premature transcript. That is, each target mRNA is implicating that introns somehow play a role in recognizing the premature mRNA targets. Studies have shown that introns located in the 5' or 3' untranslated regions play important roles in controlling NMD sensitivity of transcripts. In mRNA transport of chromatin assembly, it has been reported that spliced transcripts are exported faster from the nucleus to the cytoplasm than their unspliced counterparts, indicating the association between splicing mechanism and nuclear export, although there are some contradictory studies. In fact, nuclear transport to the cytoplasm of transcripts containing introns in their 5' untranslated regions was known to be regulated by the transcription export complex and the serine-arginine-rich protein, whereas the transport of transcripts lacking introns in their 5' untranslated region was regulated by signal sequences located in the open reading frame of those genes. A recent experiment using fluorescence in situ hybridization has investigated how introns bearing an intronless construct are distributed differently across the nucleus and the cytoplasm, and showed that intron-bearing transcripts are preferentially located in the cytoplasm. There are some studies suggesting that introns may have a role in chromatin assembly as well. Recent genome-wide mapping analysis of nucleosome positions have shown that nucleosomes are relatively depleted in introns region compared to exonic region. Schwartz et al. Have suggested that sequence element of introns ends may be responsible for nucleosome depletion in introns by pushing the nucleosomes away towards the exons. In the next role of the introns, we have introns' length matters in the efficiency of natural selection. According to Cameron et al., long introns are favored because they increase the efficiency of natural selection by releasing the Hill-Robertson interference. The Hill-Robertson interference was basically described as genetic linkage between two sites under selection in finite population, leading to decrease in the effectiveness of natural selection. The HR interference model depicts that selection efficiency should be different between genes that differ in exon-intron structure, so that genes with longer introns should be under weaker HR interference by increasing recombination between the two sites in two neighboring exons. In other words, introns may have a role in relaxing intragenic HR interference between sites under the influence of natural selection in finite population. Recombination gives the opportunity for two independently occurring favorable alleles at linked loci to be located together and thus enhance the efficiency of natural selection, which can be one of the plausible scenarios of how introns have sustained through evolutionary history of genes. Introns can also provide a source of new genes. Recently, Carvunis et al. Suggested a very interesting hypothesis about how novel genes arise from non-functional translated open reading frames named protogenes by showing that hundreds of short open reading frames of protogenes located in non-genic sequence were actually translated and might provide adaptive potential to cells in different physiological environment in Ascomycetes phylogeny, including Saccharomyces cerevisiae. According to their model, the short open reading frames can evolve into a real functional gene through a kind of continuous evolutionary process. In that sense, long non-coding intron region in higher eukaryotes can be a good reservoir for short and non-functional open reading frames. Threads-associated single nucleotide polymorphisms are also enriched in introns. Genome-wide association studies compare the allele frequencies of case groups, that is disease groups, and control groups, that is the normal groups of study participants, to identify the single nucleotide polymorphisms that are significantly more enriched in case groups than control groups. If an allele is significantly more frequent in case groups, the allele is said to be disease-associated allele or a thread-associated single nucleotide polymorphism referred to as TAS. In theory, TAS are considered to reside near sites of actual disease-causing mutations in genomes. Interestingly, most of the TAS detected by genome-wide association studies have been mapped to intron region rather than exonic or non-synonymous sites. After a discussion about introns and exons, we will now move to the discussion about reverse transcription. In introduction, reverse transcription is the process of synthesis of a complementary DNA referred to as CDNA from an mRNA template by the use of an enzyme called reverse transcriptase. It is an inherent property of retroviruses. Reverse transcription begins when the viral particle enters the cytoplasm of a target cell. The viral RNA genome enters the cytoplasm as part of a nucleoprotein complex that has not been well characterized. The process of reverse transcription generates in the cytoplasm a linear DNA duplex via an intricate series of steps. This DNA is collinear with the RNA template, but it contains terminal duplications known as the long terminal repeats or LTRs that are not present in viral RNA. Extant models for reverse transcription propose that two specialized template switches known as strand transfer reactions or jumps are required to generate the LTRs. Now let us look into the process of reverse transcription. Retroviral DNA synthesis is absolutely dependent on the two distinct enzymatic activities of reverse transcriptase, which is a DNA polymerase that can use either RNA or DNA as a template and a nuclease termed ribonuclease H or referred to as RNase H, that is specific for the RNA strand of RNA DNA duplexes. Although a role of other protein cannot be ruled out, and it is likely that certain viral proteins, as for example nucleocapsids, increases the efficiency of reverse transcription, all of the enzymatic functions required to complete the series of steps involved in the generation of a retroviral DNA can be attributed to either the DNA polymerase or the RNase H. The process of retroviral DNA synthesis is believed to follow the following steps. In the first step, minus-strand DNA synthesis is initiated using the 3' end of a partially unwound transfer RNA, which is annealed to the primer binding site in genomic RNA as a primer. Minus-strand DNA synthesis proceeds until the 5' end of the genomic RNA is reached, generating a DNA intermediate of discrete length termed minus-strand strong stop DNA. Since the binding site of the tRNA primer is near the 5' end of the viral RNA, SSS DNA is relatively short on the order of 100 to 150 bases. In the second step, following RNase H mediated degradation of the RNA strand of the RNA SSS DNA duplex, the first strand transfer causes SSS DNA to be annealed to the 3' end of the viral genomic RNA. This transfer is mediated by identical sequences known as the repeated or R sequences, which are present at the 5' and the 3' ends of the RNA genome. The 3' end of the SSS DNA was copied from the R sequence at the 5' end of the viral genome and therefore contains sequences complementary to R. After the RNA template has been removed, SSS DNA can anneal to the R sequences at the 3' end of the RNA genome. The annealing reaction appears to be facilitated by the NC. Once the SSS DNA has been transferred to the 3' R segment on viral RNA, minus-strand DNA synthesis is resumed, accompanied by RNase H digestion of the template strand. This degradation is not complete, however. The RNA genome contains a short polypurine tract called PPT that is relatively resistant to RNase H degradation. A defined RNA segment derived from the PPT primes plus-strand DNA synthesis. Plus-strand synthesis is halted after a portion of the primer tRNA is reverse transcribed, yielding a DNA called plus-strand strong stop DNA or plus SSS DNA. Although all strands of retroviruses generate a defined plus-strand primer for the PPT, some viruses generate additional plus-strand primers from the RNA genome. In the next step, RNase H removes the primer tRNA, exposing sequences in the plus SSS DNA that are complementary to sequences at or near the 3' end of plus-strand DNA. Sixth step, annealing of the complementary PBS segment in the plus SSS DNA and minus-strand DNA constitutes the second strand transfer. In the seventh step, plus and minus strand synthesis are then completed with the plus and minus strand of DNA each serving as a template for the other strand. In conclusion, reverse transcription allows propagation of retroviruses through utilization of host cellular machinery. It is a means of recombination through retrotransposons as well as an essential process of generating intron-less gene transcript for production of recombinant proteins. The existence of introns in genome is a real mystery given the expensive energy cost for a cell to pay for copying the entire length of several introns in a gene and excising them at the exact position controlled by big RNA and protein complexes after transcription. Nevertheless, most completely genomes of eukaryotic cells so far carry introns in their genomes, and some studies have showed that introns had been propagated during eukaryotic lineage evolution. The origin of spliceosomal introns in eukaryotic lineage has been attempted to be explained by massive invasion of group 2 self-splicing introns from bacteria to eukaryotes. It is very hard to understand how and why introns propagate in eukaryotic lineage and what the beneficial effect of introns on cell survival is. Taken together, introns are clearly not junk, and they provide selective advantages to cells to be evolutionarily maintained. Nevertheless, it has expensive energetic costs. New advanced molecular biology technique will lead to functional territories of introns in a more detailed scale in the near future.